Hello everyone! For today's video, we're going to learn about protozoan disease that present in animals, especially in chicken. Coccidiosis is a common protozoan disease in domestic birds and other fowl, characterized by enteritis and bloody diarrhea. The intestinal tract is affected with the exception of the renal coccidiosis in geese. Clinically, bloody feces, ruffled feathers, anemia, reduced head size, and somnolence are observed. Coccidiosis is universally most commonly in young animals housed or confined in small areas contaminated with oocysts. Coccidia are opportunistic pathogenic. If pathogenic, their virulence may be influenced by various stressors.
Etiology and epidemiology of Coccidiosis. Emeria and Isospora typically require only one host in which to complete their life cycles. Some species of Isospora have facultative intermediate host, and a new genus name, Cystoisospora, has been proposed for this species of Isospora. Coccidia are host-specific, and there is no cross-immunity between species of Coccidia. The area around the vent is stained with blood. The infection is realized by a facial oral route. After ingestion of sporulated oocysts, sporozoites are released that enter asexual and sexual cycles of development, resulting in the emergence of thousands of new oocysts in the intestines. Oocysts are distributed by feces. Soon they sporulate and become infective for chicken. Pathoanatomically, dehydration and a high degree of anemia of the body and viscera are discovered.
Pathogenesis. Infection results from ingestion of infective oocysts. Oocysts enter the environment in the feces of an infected host, but oocysts of Emeria and Isospora are sporulated and therefore not infective when passed in the feces. Under favorable conditions of oxygen, humidity, and temperature, oocysts sporulate and become infective in several days. During sporulation, the amorphous protoplasm develops into small bodies or sporozoites within secondary cysts, sporocysts in the oocyst. In Isospora spp, the sporulated oocyst has two sporocysts, each containing 4 sporozoites. When the sporulated oocyst is ingested by a susceptible animal, the sporozoites escape from the oocyst, invade the intestinal mucosa or epithelial cells in other locations, and develop intracellularly merons. Each nucleus develops into an infective body called merozoite. Merozoites enter new cells and repeat the process. After a variable number of asexual generations, merozoites develop into either macrogametocysts or microgametocysts. These produce a single macrogamete or a number of microgametes in a host cell. After being fertilized by a microgamete, the macrogamete develops into an oocyst. The oocysts have resistant walls and are discharged and sporulated in the feces. Oocysts do not survive well at temperatures below 30 degrees Celsius or above 40 degrees Celsius. Of the numerous species of Emeria or Isospora that can infect a particular host, not all are pathogenic. Concurrent infections with 2 or more species, some of which may not normally be considered pathogenic, also influence clinical disease. Within pathogenic species, strains may vary in virulence.
Clinical findings of coccidiosis. Anemic appearance of internal organs. The wet litter and the heat in premises favored of the sporulations and therefore the outbreak of coccidiosis. Depending on the localization of lesions in intestines, the coccidiosis are divided into caecal, induced by E. tenella, and small intestinal induced by A. cervulina. E. brunate, E. maxima, E. mitis, E. mivati, E. nicatrix, E. precox, and E. nagani. In caecal coccidiosis, a marked typhlitis is present and hemorrhage are seen through the intestinal wall. Small intestinal coccidiosis, depending on the Emeria species, hemorrhage with various intensities in different parts along the intestine are observed. In many instances, the hemorrhage are petechial and could be seen through the intestinal wall.
Diagnosis of coccidiosis. Histologically, Emeria organisms at a various stage of development are detected in the epithelial intestinal cells. The diagnosis is made upon the results of a complex evaluation of the clinical picture, the microscopic lesions, imprint preparations, histological study, and flotation. Coccidiosis should be differentiated from NE, UE, and histomonosis or typhlohepatitis.
Prevention and treatment.
Treatment. Sulfonamides are widely used, sulfadimethoxin, sulfaquinoxaline, sulfamethoxin, but they should not be used in layer hits. The supplementation of vitamins A and K promotes the recovery. Prevention. First, the use of coccidiostatic. Second, immunization against coccidiosis. Third, sanitation. Fourth, feeding practices. Fifth, good management.